Today, an FDA approval in cutaneous T-cell lymphoma, a breakthrough therapy designation in colorectal cancer, orphan drug status in small cell lung cancer, rolling submission of a new drug application in multiple myeloma, and a European approval in melanoma. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved mogamulizumab KPKC, known by the trade name Poteligeo, for the treatment of patients with cutaneous T-cell lymphoma who have received at least one prior systemic therapy. The decision is specific to patients with mycosis fungoides or Sézary syndrome, which are two subtypes of CTCL. The FDA based its decision on findings from the Phase 3 Maverick study, which showed that mogamulizumab reduced the risk of progression or death by 47% versus vorinostat in previously treated patients. Results also showed that the investigator-assessed median PFS was 7.7 months in the mogamulizumab arm versus 3.1 months in the vorinostat arm. By independent review, the median PFS was 6.7 versus 3.8 months respectively. The PFS benefit with mogamulizumab was observed across patient subgroups. Moreover, the overall response rate was 28% with mogamulizumab versus 4.8% with vorinostat. Among patients with MF and SS, the ORR was 21.0% versus 7.1% and 37.0% versus 2.3% respectively. Mogamulizumab also improved ORR in patients with stage 3 disease at 22.7% versus 0 and stage 4 disease at 36.5% versus 3.1%. Among those assigned to vorinostat who crossed over to mogamulizumab, the ORR was 30.1%. In colorectal cancer, the FDA has granted a breakthrough therapy designation to the combination of encorafenib, binimetinib, and cetuximab for the treatment of patients with BRAF V600E mutant metastatic disease following one or two prior lines of treatment in the metastatic setting. The designation is based on data from the safety lead-in phase of the ongoing randomized Phase 3 Beacon CRC trial. Results demonstrated a confirmed overall response rate of 48% and a one-year overall survival rate of 62% with the regimen. At 12.6 months follow-up, the median OS had not yet been reached. The median progression-free survival was 8 months and was consistent regardless of whether patients had received one or two prior treatment lines. Among patients who had received only one prior line of therapy, the ORR was 62%. Based on the positive data from the safety lead-in phase, enrollment in the randomized portion of Beacon CRC is ongoing. In this part of the Phase 3 study, patients are randomized to encorafenib, binimetinib plus cetuximab, encorafenib, cetuximab, or investigator's choice of either irinotecan, cetuximab, or FOLFIRI, cetuximab. The FDA has granted lurbinectedin an orphan drug designation for the treatment of patients with small cell lung cancer. Data with the murine-derived treatment were presented at the 2018 ASCO annual meeting for an SCLC cohort from a Phase 2 multi-center basket trial examining lurbinectedin across various solid tumors. The overall response rate was 39.3% in this SCLC cohort. An additional 7 patients had stable disease for longer than or equal to 4 months for a clinical benefit rate of 50.8%, and another 14 patients had stable disease for less than 4 months for a disease control rate of 73.8%. The median duration of response was 6.2 months. The median PFS was 4.1 months. The PFS rate was 51.1% at 4 months and 36.3% at 6 months. The median OS was 11.8 months, and the OS rate was 79.3% at 6 months and 43.1% at 12 months. The Orphan Drug Program is intended to facilitate the development and review of novel therapies for the treatment of patients with rare diseases or conditions that affect fewer than 200,000 people in the United States. In multiple myeloma, a rolling submission of an FDA new drug application was submitted for selinexor for the treatment of patients with penta-refractory multiple myeloma. The NDA is based on data from Part 2 of the Phase 2b STORM trial, in which the XPO1 inhibitor induced an overall response rate of 25.4% in patients with penta-refractory multiple myeloma. There were 2 complete responses and 29 partial responses or very good partial responses, and the median duration of response was 4.4 months. Karyopharm intends to present full data from the Phase 2b STORM trial at an upcoming oncology conference. Previously reported data from Part 1 of the study showed that selinexor achieved an ORR of 20.5% in 78 patients with quad or pentarefractory myeloma. The European Commission has approved nivolumab as an adjuvant treatment for adult patients with completely resected melanoma with lymph node involvement or metastatic disease, regardless of BRAF mutation status. The approval was based on results from the randomized Phase 3 CheckMate 238 trial, in which the recurrence-free survival rate at 18 months with nivolumab was 66.4% versus 52.7% for ipilimumab in patients with stage 3B or C or 4 disease. There was a 35% reduction in the risk of recurrence or death with the PD-1 inhibitor versus the CTLA-4 inhibitor. Moreover, data showed that the median RFS had not yet been reached in either arm of the trial. The 12-month RFS rate with nivolumab was 70.5% versus 60.8% in the ipilimumab group. The median distant metastasis-free survival was not reached in either treatment group, with fewer events noted in the nivolumab arm. This week, we sat down with Dr. David Ilsen of Memorial Sloan Kettering Cancer Center to discuss molecular testing in metastatic gastric and gastroesophageal junction cancer.
Yeah, at the current time, if you're dealing with a locally advanced patient, molecular testing does not play a clear role. It really does not impact on choice of therapy. For metastatic disease, on the other hand, molecular testing does have an impact. Although we are in the era of sort of whole exome sequencing and next generation sequencing of cancers, it still does play a limited role in identifying treatments for gastric cancer. The main one that is standard is HER2 testing. In metastatic disease, if a patient's HER2 positive, we include trastuzumab as part of chemotherapy. And now we have to consider in metastatic disease either MSI or DNA mismatch repair protein testing or PD-L1 testing. We now have evidence that anti-PD-1 drugs, particularly pembrolizumab, has a signal of activity in PD-L1 positive gastric cancers and refractory disease, and has even a more significant activity in MSI high or DNA mismatch repair deficient gastric cancers. So in the patients up front, we do HER2 testing that impacts on initial treatment of metastatic disease. When they become refractory to chemotherapy, we now have regulatory approval of pembrolizumab for either PD-L1 positive or MSI high gastric cancer. In terms of everything else that we see in genomic sequencing, it's interesting and it's a research question, and maybe we can identify in 1% of patients some mutation that they might be an appropriate candidate for a trial. But typically we find P53 mutations, we find amplification of receptor-associated tyrosine kinases, of which the only relevant one is HER2. So although genomic testing is becoming part of a standard practice, in reality there are really three things that we need to test for: HER2, MSI, and PD-L1. Where we suspect gastric cancer in a young patient, so typically any patient under the age of 50, or when we see a significant history of gastric cancer in the family, we should think about CDH1 or familial hereditary diffuse gastric cancer. That's not a subtle presentation in a family. Typically, these are patients that develop gastric cancer in their 30s and 40s. We see it generation after generation. So just because someone's under the age of 50 doesn't mean they have CDH1 mutation gastric cancer. There's also an association with lobular breast cancer. So that's not routinely tested for. We do have consideration for doing genetic testing and counseling in any patient that develops gastric cancer under the age of 40 to 50. But usually we're not going to find a heritable mutation unless there's another family history. And CDH1 gastric cancer is relatively rare. Other rare familial gastric cancer syndromes include Lynch syndrome, where we see the association with colorectal cancer and GYN cancers in women. BRCA, breast cancer, has an associated increased risk in gastric cancer. And then we talked about non-polyposis Lynch syndrome, also the polyposis syndromes. Peutz-Jeghers, juvenile polyposis, FAP, all have associated increased risks in developing gastric cancer. These are all rare. So typically we would consider genetic testing in a young patient, or if there's a strong family history, but even in a young patient we're not likely to find a mutation. There is a situation occasionally where that patient is the index member of the family. So young patients we do test. Older patients we would test only in the context of a strong family history.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.